Well, we have about five after, so I think we're going to go ahead and get started. It is my pleasure to welcome you all to the Beef webinar series. And tonight, I'm not sure about you guys, but I've personally been looking forward to our presentation tonight for quite a while. And if you've had me in your county, you know that I have been anxious to hear Dr. Fontes and his work on bull nutrition and bull fertility. Tonight, from the University of Georgia, we have Dr. Pedro Fontes. Dr. Fontes is a native of Brazil and grew up on a large commercial cow-calf operation down there. After he got his DVM, he attended the University of Florida and got his master's, I believe, with Dr. Lamb, right?
That's
And
right. That's right.
followed Cliff to Texas A&M. And for those of you that don't know Cliff Lamb, Cliff was one of the better reproductive physiologists, and in particular, one of the better trainers that we've had in reproductive physiology probably in the last 3 or 4 decades. But Pedro has done a tremendous amount of work on both male and female and has some really, really unique work to share with us tonight on some revelations that he has had there on the impact of bull nutrition on semen quality. And just to give you an idea of where Dr. Fontes is in the world of reproductive physiology, he is among the top two or three physiologists in the country. And so it's absolutely our pleasure to have him here tonight and my pleasure to introduce Dr. Pedro Fontes. And we are really happy that you're here with us tonight.
Great. Thanks, Dr. Anderson. I appreciate the nice introduction. Not sure about the ranking there, top 2 or 3. I'm not sure if I'm ranked that high, but I appreciate the opportunity to share some of our work. Just want to double check. Are you seeing my screen correctly, or you're seeing because it changed here on my end?
It looks good to me.
Okay, because it changed here on my end, but I can still see it here. All right, perfect. Thanks again, Dr. Anderson, for the introduction, and yeah, for the opportunity to share some of the work, like you mentioned, that we've been doing on the male side of things. As you mentioned, I was trained by Dr. Lamb, and Dr. Lamb, program historically has mostly focused on the female side of things, and that's where my training was focused as well. But since I, finished my graduate work at Texas A&M and had the opportunity to start in my current position here at the University of Georgia, I have a pretty heavy extension appointment. And here in Georgia, one of the really staple extension programs that we have is our Georgia Bull Evaluation Program. And that program is led by Jason Duggan and Dr. Stewart. Now we recently recruited Dr. Carvalho that is going to be leading the Tifton location. But that program is really nice because we get to interact not only with the seed stock producers that are consigning the bulls, but also with the commercial producers that come and purchase these bulls at the end of the bull. At the end of the bull test, when we organize a sale with the sale eligible bulls, and through that program, I had the opportunity to interact quite a bit with commercial producers and hear some of their concerns. And one of the things I really like about our program is that, our bulls, they're not extremely overconditioned. But that's one of the things that, we work really hard to do to accomplish, to make sure that those bulls look good in a sale, but they're not extremely overconditioned. But we know very often beef bulls do get overconditioned out there. And through that interaction, I started to switch gears and focus not only on the female. We still do quite a lot of work in the female side of things, but try to understand a little bit of that overconditioning aspects on male fertility. And that's what I'm going to be focusing my presentation today. So over the next 35 to 40 minutes here, what I'm going to do is first try to give you guys an overview of some of the observational studies that we have done, trying to see if there is a potential relationship between overconditioning and bull fertility. And then I'm going to switch gears and go over some of the control studies, not only done by our group, but also some other groups out there that have been doing some studying of on bull overnutrition. And then towards the second half, probably last third of my presentation here, I'm going to spend some time in some of the more recent work that we've been doing, where we're not only looking at the impact of overconditioning on semen quality itself, but we're actually looking at the ability of that semen to fertilize an egg or an oocyte and hopefully produce an embryo. So I'm going to share some of that recent work with you guys today as well. So getting started here, looking into some of our observational studies. So this is a study, the study that I'm going to share with you now. It's a study that my graduate student, her name is Molly Smith. Basically, what Molly did here in this study, she compiled data from 3 different consignor-based bull development programs in the Southeast United States. And basically what she did is she explored the relationship between the amount of subcutaneous back fat thickness that those bulls had based on a carcass ultrasound. Okay. And those are young yearling bulls around 15 months of age. And she correlated that information with the actual breeding soundness exam outputs. And what she was trying to ask here or evaluate here is, well, is there an association between subcutaneous back fat thickness and any of the semen parameters that we look at in a BSE? Right, so here's a distribution of the bulls according to their back fat thickness. And again, this is based on an ultrasound exam, okay? A carcass ultrasound exam here. And you can see that, our bulls follow what we call a normal distribution here. And what we did in this study here is basically we separated our bull population into the amount based on the amount of subcutaneous back fat that those bulls had. So we separated these bulls based on the bulls that ranked in the top 10%, the bulls that rank in the bottom 10%, and the bulls that fell somewhat in the middle here, representing about 80% of the bulls in the study. And as I shared this data here, I wanted to keep in mind that this is phenotypic back fat thickness. Okay, that's not the back fat EPD that you see in a sale catalog. That's the actual EPD that we're measuring in the ultrasound exam, not the genetic merit for back fat thickness, but actual the phenotypic back fat thickness that those bulls had. And we're lucky enough that we're able to leverage over 700 breeding soundness examination records here in this study to try to explore the relationship. And what was neat that Molly showed is that the bulls that were in the top 10% of the population, so if you look here in this figure on the left, you can see that they had significantly fewer cells that were classified as normal. Okay, and this is basically on a morphology evaluation of the semen, where basically a veterinarian is going to be looking at how well packed together those cells are, right? And you can see that a fewer percentage of the cells were normal in the bulls that were in the top 10% for back fat thickness. When we break the abnormalities down into primary and secondary abnormalities, you can see that for both primary and secondary abnormalities, the bulls that were in the top 10% of the population, they had a greater percentage of cells showing both primary and secondary abnormalities. And what's neat is if you look at these numbers here, there's not a whole lot of difference, even though, we go there and say, hey, this is statistically significant, right? But it's only about a 7% difference here. Then the natural question here is like, well, but does that influence the ability of those bulls to actually pass a BSE? And this is what Molly showed here, that in fact it does. So the bulls are in the top 10% of that population for subcutaneous back fat thickness. So again, those are the more overconditioned bulls based on how thick their back fat was. You can see that a larger percentage of those bulls fail their breeding soundness exam when you compare to their counterparts here that were a little bit linear. If we get this data here and we just plot it a little bit different here we have the probability of the bulls failing their breeding soundness exam versus the subcutaneous back fat thickness. And I apologize, this is in centimeters here. But basically, as you increase subcutaneous back fat thickness, you increase the probability of bulls failing the BSC. And you can start seeing a more clear increase here at 1.2 centimeters. So if we convert that to inches, that's going to be at about half an inch, 0.55 inches here. So when those bulls start having a little bit more than half an inch of subcutaneous back fat thickness, that's when we start seeing a more clear increase in the probability of them actually failing the BSC. So pretty interesting. So if you go out there in a population of bulls and we sort the ones that are a little bit more over-conditioned based on an objective metric, it seems like we can show some differences in semen parameters, even though those bulls are managed pretty much the same way, right? They're all in a consignor-based programs. They're all were brought to 3 specific locations and managed in those 3 specific locations. Even though we're managing them the same, if they're getting a little bit more over condition, they're more likely to have, it appears that they're more likely to have some fertility issues there. So we have a few months back recorded a podcast, and this is a picture of Molly here. So if you're driving down the road and you're bored, you're welcome to scan this QR code here and save this episode. So we recorded an episode in which Molly goes a little bit more in depth into this work that she's done in this observational study looking at the relationship between back fat thickness and semen characteristics. And, you know, I guess as a scientist, I think that the natural thing, if we go out there and we see something in the field, you know, producers are concerned, we collect some data in the field, and then we see those same patterns. It seems like the concerns that those commercial producers are bringing to us, they're relevant, right? We do seem to see that association between back fat thickness in those bulls, in young beef bulls, and semen characteristics. The natural next step for us is like, all right, let's bring this into a control environment in the university and try to study that a little bit more, right? So can we experimentally get bulls over condition, and by doing that, can we actively induce changes in semen characteristics in experimental control settings? And what's interesting is that if you look in the literature, there's quite a lot of work coming out of Canada exploring that. These work was predominantly performed in the 90s, late 90s, where basically they went there and experimentally fed bulls either a high concentrate diet, I'm calling it here a high gain diet, or a controlled diet. And you can see that those bulls, after being fed for about 170 days here, 168 days, the bulls that were fed the high concentrate diet, they had greater scrotal circumference. But not only that, they had a decrease in sperm motility and an increase in sperm morphology defects. In this specific study, they saw more secondary defects. One of the neat things they did in the study is that they went there with infrared thermal cameras and they measured the temperature of the scrotum, the surface temperature of the scrotum of these bulls. And they noticed that the bulls that were in the high concentrate diet, so in the high gain treatment here, they had a bit of an impaired ability to cool their testes. So when they compared the temperature from the top to the bottom of the testes, they noticed that those bulls, they were not cooling the scrotal surface temperatures they were supposed to when they were exposed to this high energy diet. Okay. And that's interesting because that's one of the big hypotheses that we have by which we think those bulls that get overconditioned might be potentially having suboptimal sperm production or suboptimal semen quality. Choose the correct term here. So this is a picture here on the left from a study that we had the opportunity of harvest bulls that we purposefully overcondition. And what you're seeing here, you have 2 testes here. This is the reproductive system or the reproductive tract of those bulls. And then here in the middle, this is the penis of the bull here. And I want to draw attention to the amount of fat accumulated in what we call the pampiniform plexus region, okay? Pampiniform plexus sounds like a fancy, big, complicated name, but all it is, it's a cooling system that bulls and matter of fact, humans have as well in which we get in which the testis is able to cool the blood that it's coming from our abdominal cavity into the testis down before that blood reaches the testes. And the reason why that happens is because sperm production happens in a little bit of a cooler temperature. And what we know is that if we go there and purposefully get the scrotum of those bulls warm, and we can do that by putting legally a scrotal jacket, we do like a little scrotal insulation there, we really decrease sperm production when we do that. So one of our hypotheses here is that because those bulls accumulate fat right over the system, this cooling system here, that they have in what they call the spermatic cord here, we end up limiting the ability of those bulls to cool the testes down and promote adequate spermatogenesis. And as a consequence of that, we see some changes in fertility. When I present this, usually one of the questions that I get, mainly on the seed stock producer side of things, is like, "Hey Pedro, but if I slow down gain on those bulls after weaning, I might have some issues with those bulls not reaching puberty." And that's a good point. If those bulls are in a very suboptimal plane of nutrition, as a matter of fact, they will have delayed puberty achievement. But what's interesting is that it seems like the timing in which those bulls have a lower plane of nutrition matters when it comes to whether they reach puberty early, when they're around yearlings, 12, 13 months of age, or not. So this is a study with Holstein bulls, and don't hold it against me that this is a study using Holstein bulls. The reason why this is good is because, you know, when they're developing these Holstein bulls, they can really dial down the diet, right? Because those Holstein bulls, they're weaned at birth. So experimentally, that's really good because scientists can go there and really control how much those bulls are gaining. And this is a study out of Ireland where they basically had these Holstein bulls in 2 different planes of nutrition during the first 6 months of life. So you can think of it as you know, the phase in which those bulls, if they were, you know, your traditional growing beef bull, they will be nursing, right? At this stage, this will be pre-weaning. You can think of it this way. So they're either fed to gain 2.6 pounds a day or 1.1 pound a day in the low treatment versus the high treatment here. And then within each one of these treatments during their first 6 months of life, in the second 6 months of life of these calves or these Holstein bulls, they're either fed to gain 3.5 pounds, or fed to gain 1.5 to 2 pounds here in the low gain treatment. So they had what we call a 2x2 factorial treatment, where you have the different combinations of a high plane of nutrition or a low plane of nutrition early in life, high plane of nutrition, low plane of nutrition after weaning, right? And what was neat is that in this study, they didn't see any differences in age of puberty or age in which those bulls reach sexual maturation based on the post-weaning diet, okay, which is quite interesting. So in other words, basically the main driver of age at puberty in this specific study was in fact the diet in which those young bulls were exposed during the first 6 months of their lives, rather than the second 6 months of their lives, right? And that's interesting, mainly if you are involved in a cooperator type of program. So if you are in the seed stock business, you might be putting embryos in a commercial cow-calf herd. You know, this is important because the nutritional plane, not only of that cow during gestation, but also the ability of those commercial cows to nurse those purebred young bulls is going to drive their ability to potentially reach puberty or not in time to pass a breeding soundness exam before those bulls are marketed, right? So there's some direct applications for us here in the beef industry. The other neat thing about overconditioned bulls, about the idea of overconditioning bulls, is that as you start studying overconditioning, right, there's probably fewer mammals out there that are as good as humans at getting overconditioned. So in the human fertility side of things, studying overnutrition and fertility and male fertility is something very important, right? And when you browse through the male literature, through the human literature on the relationship between male obesity and fertility, there's a lot of things that resemble what we see in bulls, So in humans, we also see increased scrotal temperature, altered sperm parameters. But one of the really neat things that we see in humans when we have in situations of obesity is a decrease in embryo production. And I had no idea that that was the case, right? So very often when couples are struggling to conceive, right, the doctor, the fertility doctor will send them through an IVF round to try to generate a pregnancy through IVF. And what they notice, and this is what this study is showing here, this is human fertility clinic, and I promise I'm coming back to beef bulls in a minute, but this is data, epidemiological data in human fertility clinics evaluating the relationship between body mass index, which is an estimate of body adiposity, right, and IVF outcomes in those human fertility clinics. And basically, when you have a BMI or a body mass index above 20 weight, you're classified as being obese or overweight. And when you compare the couples where the male was overweight, you can see that that's the gray bars here. You can see that these couples had a lesser percentage of fertilization rates. Not only that, the percentage of transferable embryos that they generated was also less if the male was overweight. And what's even more interesting, whenever they went there and transferred those embryos, their probability of generating a clinical pregnancy was also decreased if the couple if the male was obese, when they collected semen to go through the IVF process in these human fertility clinics. So seeing that data, the natural question is, well, does that happen in our beef bulls, right? So we then started to hypothesize that, well, maybe when we get those young beef bulls over conditioned, we're going to induce a series of metabolic changes in these bulls. And as a consequence of that, we're going to change sperm quality, right? We're going to induce some specific changes in sperm. And those changes in sperm quality not only is going to influence the ability of those sperm cells to swim and fertilize the egg, but also potentially the ability of the embryo, once it's fertilized, once that egg is fertilized, once the oocyte is fertilized, it might influence the ability of those oocytes to continue to develop. So historically, we always thought about males, the sperm cell as its role being, swimming through the female reproductive tract and fertilizing the egg, and that was it, right? Depositing its DNA there, then its job is done. But the reality is, what we're learning is that the bull plays a big role, particularly in early embryonic development. And there's a lot of early embryonic mortality that is actually driven by male-related factors. So with that, we wanted to test if that was the case in overconditioned bulls. And to start diving into that, we first did a study with mature bulls. So we got bulls from our commercial herd. Those were purebred Angus bulls that we use as cleanup bulls in one of our UJ commercial herds. And when those bulls are off season, we randomly assign 8 bulls to either a diet where those bulls were gaining about 4 pounds a day, or a diet in which those mature bulls were fed to maintain their body weight. We call it a maintenance treatment here. And those bulls were fed their respective diets for about 67 days. And the reason for that is because a spermatogenic cycle takes 60 days in bulls, approximately 60 days in bulls. So we want to make sure that those bulls are exposed to that diet for at least 60 days. And then after we fed those bulls for 60 days, we collected semen from them, and we sent them to our colleagues at the University of Florida, at John Bromfield Leddy's in vitro embryo production here, and basically using semen from bulls that were either exposed to the high gain diet or the maintenance diet. What's interesting is that in this first study, we didn't see any differences in cleavage rate. However, when we look at the percentage of oocytes that actually become what we call a blastocyst, that's a 7 days old embryo, okay? You can see that whenever we use semen from the high gain bulls, we had a lesser percentage of or a decrease in blastocyst rate, both as it relates to the total number of oocytes that were included, as well as the number of oocytes that were cleaved. Basically, what this means is that the sperm likely fertilized the embryo, it started cleaving, right? That's a cleaved structure. So out of those structures that cleaved, a fewer percentage of them actually make it all the way to what we call a blastocyst, which is a 7 days old embryo, showing us that maybe those bulls that get overconditioned were not only influencing semen quality, but we might be influencing the ability of those embryos to become viable embryos and potentially generate a pregnancy. But the reality is, this was done in mature bulls, right? If you think about going there and getting a mature bull and getting that bull to gain 4 pounds a day, guess what? That bull is not going to put on a whole lot of skeletal muscle tissue. It's going to be predominantly be putting on fat. So the question then is, well, does that happen in our population of bulls that usually get overconditioned, which is our young bulls that might be going through a sale, right? So we then try to replicate that study in young bulls. And the way we did that, we got half siblings bulls that were all sired by the same sire. Okay, there was 44 bulls in the study. And then we randomly assigned them to either a moderate gain diet, where those bulls, and I apologize, this is in kilograms here, but those bulls were fed a diet to either gain 2.7 pounds a day. That was the moderate gain target average daily gain. And then for a high gain treatment, the target average daily gain was 4 pounds a day. Okay, so again, 2.7 versus 4 pounds a day average daily gain. And those bulls were fed a very similar diet from an ingredient standpoint. In other words, the ingredients were the same, but the amount of ingredients changed in order to increase the caloric density of the diet in the high gain group. And then we fed them for 114 days, and this is a diagram of what we did. Basically, we adapted those bulls for about 7 days. Those bulls were already bunk broken when we started this study. And then we put them in a system where we can monitor their individual feed intake, and we fed them for, like I said, 114 days. And throughout the whole feeding period, we will go there, collect body weight, body condition score, collect some blood samples from those bulls, and we also scan them with a carcass ultrasound to see if they were depositing fat as we wanted them to. Then at the end of the study, we go there and we collected these bulls 3 times. So what we do, we go there, do what we call a cleanup collection. So we get rid of that old sperm that is stored in the epididymis of the bull, and then we collect them 3 times. And the reason why we collect 3 times is because there's a little bit of variation between collections, and we're going to paint a true picture of the fertility of the bull. So we go there and try to collect them several times to dilute the effect of collection there and get a more representative sample from each bull. And then we do not only our traditional breeding soundness exam, but we use some more advanced techniques that I'm going to show you here. One of them is CASA, so that stands for computer-assisted sperm analysis, and the other one is flow cytometry. And I'm going to explain those techniques real quick, but basically we collected the semen from these bulls, we evaluated shoot side, evaluated morphology, and then we went there and extended the semen. We froze some of that semen. And then part of it, we sent you those more fancy analysis. So where we have a computer analyzing the sperm, so it's not a subjective analysis, and a flow cytometry technology where we basically, instead of looking at 100 cells like we normally do in a morphology assessment, we'll look at anywhere between 10 to 20,000 cells here to paint a better picture. We use some new technology here to paint a better picture of the characteristics of the sperm of those bulls. And then going to the results, you can see that we did a pretty decent job getting a different plane of nutrition. Both groups gain a little bit more than we were expecting, but you can see that the high gain bulls gain significantly more than the moderate gain bulls during the feeding period. But what was interesting is when we look at body condition score of those bulls, you can see that at the end of the feeding period, there was a clear separation between the moderate gain and the high gain. What was interesting is, and I apologize, this is in centimeters here, but those bulls that were in the high gain treatment at the end of the feeding period, okay, they had about 1/2 inch on average of subcutaneous back fat thickness based on the carcass ultrasound. So basically, this is the exact same as the bulls that were in the top 10% of that population for that initial observational study that I showed you. So we were able to generate the exact phenotype that we wanted, right? We're able to exactly get those bulls where we wanted them to really get them over condition. And what was cool was when we look at those bulls, I'm showing you some insulin to glucose ratio data, okay? And don't get you caught on that, but basically what this data is showing is that those bulls became what we call insulin resistant. And insulin resistance is something that we see in human obesity as well, right? Whenever we are in a situation of obesity, you hear about people developing type 2 diabetes. Well, those bulls were not type 2 diabetic, but they had insulin resistance, which is something that we see in people that have type 2 diabetes is associated with bad diet habits. And what's interesting is that those bulls have an upregulation of some pro-inflammatory markers. So haptoglobin is something that it's a protein that is produced by the liver as well as the adipose tissue, and it's produced when animals are in conditions of stress. And you can see that the bulls that were in conditions of inflammation. Okay, so conditions of stress often drive an increase in haptoglobin. But not only that, when we have increased fat deposition, we see an increase in haptoglobin production, and that's what we saw in the bulls that were in the high gain treatment as well. So those bulls not only look different, but when we start measuring some blood markers, their body is functioning a little bit different there as well, right? So when we go there and look at the semen characteristics for this study, you can see that here we have total motility and progressive motility. And again, these were done using a computer-assisted sperm analysis system. This is not a subjective estimate of sperm motility. And you can see that the bulls in the high gain treatment tended to have less sperm motility. And when we use we call it we use a 60% threshold of sperm motility to consider that semen eligible for freezing. Okay, assuming that we were in a scenario where we're going to collect those bulls in a semen stud facility or potentially market semen from those bulls, you can see there are a fair proportion of the bulls as well as the fair proportion of the ejaculate of the bulls in the high gain treatment would meet motility quality standards based in our data set. So this is quite interesting if you think about applications for bulls that might be going to the semen companies and it might be struggling to be collected, mainly when they're young and sometimes a little bit overconditioned. What's interesting as well, here's the part of the data. I'm not going to bring all the data on the flow cytometry because there's a lot of data that system spits out, but I'm going to show you what I think is most interesting is this is the percentage of cells that had an intact plasma membrane, right? So the sperm cell is just like any cell in our body. They have a plasma membrane, right? And a sperm cell with a ruptured plasma membrane is basically unable to fertilize an egg in vivo, right? So the more disrupted plasma membrane we have, the worse the fertility of that bull is expected to be. So, and what we do is we look at the plasma membrane and those bulls in 2 time points. So we collect the bull and then we look at a 0 hour of in vitro capacitation, and then we put them in this what we call capacitation system, where basically we insult that sperm cell a little bit. And you can see that after we push those cells a little bit, we can see that the bulls in a high gain treatment then have a fewer percentage of cells with an intact plasma membrane, which is telling us that those cells might be a little bit more susceptible to stress in the bulls that were exposed to that high gain diet. So again, we're changing the biology of those sperm cells as well, and it doesn't seem like those changes are very positive. It seems like we're moving the biology not in the direction we want it to go. All right, so we then went there and collaborated with Dr. Brumfield again to do in vitro fertilization in those young bulls now. It was interesting. We saw some very similar results. Okay, so here you can see that the bulls that were exposed to the high gain diet, and this is your dark color bars here, they had a fewer percentage of the eggs cleaving or undergoing cleavage in the in vitro production system. Not only that, you can see that those embryos, they had a slower pattern of growth. On day 6 and day 7, a fewer percentage of oocytes actually became a blastocyst. So this is interesting because here on day 6.5, day 7, so if you send a bull, if you're in the seed stock business and you're trying to produce embryos, that's when they will pack those embryos into straws, either for freezing or for shipping to your embryologist so they can put those eggs in. And you can see that a fair percentage of the oocytes that came uh, um, that were fertilized with, that were exposed to the semen from bulls that were exposed to the high gain diet actually made it to the blastocyst stage by day 7. Not only that, we took that a step further and we use this really nice technique that we can count the number of cells in the embryos, and usually embryos with lower quality, they have fewer cells, and fair enough. The embryos that were produced with semen from overconditioned young bulls had fewer cells. Okay. But not only that, we use this similar technique to stain the cells that are undergoing apoptosis. Basically, these are your cells, the embryonic cells, they're dying. Okay. And what's pretty neat is that the embryos that were sired by the overconditioned bulls, they had more cells undergoing this process of apoptosis, which is basically a process of cell death. So they had more cells dying in the embryos that were sired by the high gain bull. And here's a representative picture of those embryos here, which is pretty interesting, right? It's not only about making it to the blastocyst stage, but how good of a quality that blastocyst really is. So whenever they're in the uterus of the cow, can they successfully generate a pregnancy? What was neat is that we put those embryos through a time-lapse imaging system, and that's what you're seeing here on the right. So we have a video of an embryo growing in the moderate gain treatment and in the high gain treatment. And you can see that the embryos that were sired by bulls in the high gain treatment, it took them a longer amount of time to reach some key stages of development. Here's the morula stage, and here's the blastocyst stage. This is a 7 days old embryo, okay? Blastocyst hatching time here, 7.5 days embryo here, which is quite interesting, right? We're not only decreasing the quality, but it appears that those embryos are growing at a slower pace. So if you look at the image here on the right, you can see that this embryo has clived multiple times, where this one here is still in what we call a 4-cell stage here. So it's lagging behind a little bit. One thing that I want to draw your attention while the time lapse is playing here, and this embryo is already becoming what we call a morula here, okay, is that the embryos that were sired by the overconditioned bulls, they also had a little bit greater cytoplasmic fragmentation as well, okay? And you can see those embryos continue to develop. You can see that the embryo on top is still in a little bit more advanced stage, and you can see start forming a balloon here. That's what we call the blastocell, okay? And that's what we call a blastocyst. And you can see that this embryo is significantly more developed and is about to start hatching here, the process of hatching. So again, through the time-lapse image, we were able to show that it takes longer for the embryo in the high gain treatment, slightly longer to develop when you during those early stages of development, when you compare to embryos sired between high gain versus low gain bulls. So just in summary here, they had decreased cleavage rate. They took longer to become a morula. This is the line showing the different stages of development. Not only that, they had more dead cells. And not only that, they took longer to hatch and start and to basically hatch from what we call the zona pellucida here. So then we decided to ask ourselves, like, well, what if we put those embryos in? So we then went through the process of generating those embryos again, but at the same time, we're generating those embryos using semen from either those overconditioned bulls or the moderate gain bulls. We went there and set up some embryo recipient cows. And what we did is we transferred 2 to 5 embryos in each one of these cows. And what we wanted to do that is put that embryo in the uterus of the cow, and then about 7.5 days later, flush those embryos out. And this is my graduate student, Lucas, here. And what he's doing is basically trying to recover those embryos out of those cows, and those embryos are 15 days old. And this is a video of one of those embryos. They look a little bit like a tooth, like a floss. if you look at the video here, you can see the little embryo right where my pointer is here. Okay. And what Lucas is doing, he's doing a series of molecular analysis of those embryos to see how well they're growing. I'm not going to bother you with that, but what I'm going to show you is one of the interesting findings that Lucas did is that he basically measured those embryos to see how well they're elongating. So usually, the faster those embryos elongate, we think that the greater the probability of that embryo to actually establish a pregnancy. Well, it's interesting when we look at the size of the embryos. Remember, we transfer multiple embryos in those embryo recipient cows, and see there was a tendency for a decrease in the size of the embryos after we transfer and we leave them into those cows, into the uterus of the recipient cows for about 7 days. So they're not only growing different in an IVF dish, but actually when we go there and put it in the embryo recipient cow, it appears that they grow at a slower rate. And what we know is that embryos that grow slower in a both in vitro system as well as they elongate less in utero, they tend to struggle more to establish pregnancy as well. So it seems like the diet, this is quite interesting, right? It seems like the diet we're feeding the bulls, our bulls, is influencing the ability of the embryos that are sired by that bull to actually grow in the uterus healthy and potentially establish a pregnancy there. So I'm around 35 to 40 minutes here, so I'm getting ready to wrap up. So just in summary here, you can see that whenever we go there and get those young bulls over condition, we alter their metabolic characteristics, right? We increase systemic markers of inflammation. Those bulls get a little bit of insulin resistance. They increase quite a bit fat deposition. That leads to some changes in semen quality. But not only that, one of the things that are important to keep in mind is that even if those changes in sperm quality, they're subtle, we change sperm biology enough to alter subsequent embryo development. So a lot of those bulls might pass a breeding soundness exam. The reality is whenever we put them to work and they have to fertilize an embryo and produce an embryo that is going to successfully hatch from the zona pellucida, elongate inside the uterus of a cow, it seems like those diets might be altering the biology of that sperm in a way that is harming these important biological processes here. So with that, I'm going to take a minute here to acknowledge all the graduate students and the farm crew that was involved in the series of studies that we did, exploring this idea of sire overconditioning, as well as the faculty members and graduate students and farm staff that collaborated in these studies. Not only that, I want to also acknowledge, of course, the funding sources that enable us to get this kind of work done and provide you with that information so you can make some informed management decisions at your place and hopefully optimize bull fertility there. So with that, Dr. Anderson, I'm going to hand it over to you. If we have time, I'll be happy to entertain any questions. Thank you very much.
Fantastic, Dr. Fontes. That was exceptional. We have one question that's already in the in the chat box. And remember, if you have any questions, just type them in. We'll answer them here. So just type them in at any time. But David Hamilton asked or mentioned that he didn't have an ultrasound. Can bulls be reliably evaluated for body condition score like cows?
Yes, yes. In fact, they can. That's a great question, David. And you're right. Most frequently, we're not going to have an ultrasound in hand, right? Unless we're scanning, yearling bulls and reporting carcass data to the breed association. We're not going to have that carcass ultrasound information in hand. So you can use a traditional body condition score system. Okay. So if you're using a 1 to 9 body condition scoring system, I'm okay with bulls getting all the way to a 6.5 there. If you are below a 7, you shouldn't see any major issues from a semen quality standpoint. We start seeing those issues when those bulls get to a 7, 7.5, 8. And very often we don't give bulls scores that high. We tend to underestimate how overconditioned they are. But if we're honest to ourselves, we know that sometimes those young bulls, mainly as they're going through a sale ring, very often they are, in fact, in a body condition score of an 8 and or a 7 and an 8. And in this situation, we know that a certain percentage of them will start having some fertility issues. But yeah, one of the things that I always try to do as well. Sometimes I scare people to death when I show this data, and one of the things I want to draw your attention is you probably noticed that it's not a train wreck, right? It's not like those bulls will get completely infertile if they are over conditioned, but a certain percentage of them will struggle to pass the BSC, and some of them will pass, and their fertility will be a little bit suboptimal. So keep that in mind. So body condition scoring goes a long way. We want those bulls. We know those bulls are going to lose condition very often during the breeding season, so we want them to have some reserves. By no means I'm telling people to have bulls, pretty lean before the beginning of the breeding season. We know they need some gross resin and they need some body reserves before we turn them out. So we usually like to see those bulls at a body condition score of a 6 before we turn them out. So they'll have a good amount of fat cover that they can burn through while they're working the breeding season. Because we know they're working hard. Sometimes those bulls will lose over 100 pounds during the breeding season. So yeah, keep that in mind.
Got a few questions for you. Um, One of the terms you've used a lot tonight, was young bulls. We don't only have young bulls.
Yes.
So I assume that this phenomenon is consistent regardless of age, but it's possible that if we're pulling bulls, and we're running about 60% year-long calving seasons down here, Doc, and so sometimes we don't always pull the bull, but when we do pull the bull and they get a little bit older and maybe they lay around a little bit, they can add some condition. I assume that this continues on throughout lifetime.
Yeah. So in one of our studies we did with mature bulls, and we saw a decrease in embryo production as well. So yes, if the mature bulls get over conditioned, they can also have similar issues there. And one of the things I didn't talk about is it's also the structural soundness component of it as well. As we're thinking about those bulls getting over conditioned early in life or even late, we know that mainly with the genetics we have today, those bulls are extremely heavy, and we put a lot of pressure into their muscle skeletal system, right? So that's something to keep in mind as well. But yeah, the mature bulls, even though it's a little bit harder to get them very over conditioned, but we know sometimes they do because they just sit around for 7, 8 months, and they can add some weight on. But yeah, if you keep an eye on them, they can have the same issues, but if you keep an eye on them, you can hopefully manage that so they don't get extremely over conditioned. One of the issues that we do have here, I don't know if you guys get a little bit of that up there in Kentucky too, but we have a lot of issues with those 18-month-old bulls that maybe they were bought at 14 months of age. They're used in a first breeding season, and then they get turned out with the mature bulls. And those bulls, they lose a lot of weight in their first breeding season. They're still growing, and sometimes we don't give them enough attention during while they're resting after their first breeding season. One of the things that I always try to tell folks is keep in mind those bulls you can think of them as first calf heifers, right? They're still developing themselves, and they need to be fed accordingly as well. We need to be mindful that their nutritional requirements might be a little bit higher on a per pound basis, um, on a weight of the bull basis. We got to keep that in mind to make sure that we allow those bulls to develop properly as well.
So I go home and or go out tomorrow and look at my bulls after this talk, and I notice my bulls are my 2 and 3 year old bulls are all quite a bit overweight. What do I do?
What do you do? Good question. Um, So you can try to get some weight off them, not a lot, mainly for turning them out of breed. If those bulls are in a predominantly forage-based diet, which most of them will be, there's not much that we recommend you to change. And the reason for that is because a negative energy balance is also not great, right? Um, what is interesting though is there's some studies where there's a really nice study from North Dakota State that they purposely got the same group of bulls and they either gain weight or they have the bulls gain weight and half of them lost weight. And the bulls that lost weight had better semen quality at the end of the study than the ones that gained weight. So it seems like losing condition is not as bad to fertility, but again, we got to be mindful, right? We don't want our bulls in a body condition score of 4.5, because then you might see some issues there. But um, yeah, I wouldn't recommend abruptly changing their nutritional program. But yeah, try to make sure they're in a predominantly roughage based diet. Whenever they're going to go out there and start working, they're going to be exposed to a predominantly roughage based diet with your cow herd anyways. So that's the recommendation that I have.
The, I mean, I know overcondition impacts libido. What does that? So, I mean, so where I'm going with this, if I had to choose between 4.5 and 5 and 6.5 and 7, I mean, if you just had to choose, my tendency would be to think we might want to do 4.5 to 5 versus 6.5 to 7. But I don't know what the impact on libido is because, I mean, they could have, the highest quality semen in the world. If they don't have enough energy, they don't have enough drive and so forth, they're not going to get cows pregnant either. So is there any, I mean, I know this isn't part of your talk tonight, but it certainly
No, yeah,
stimulated me to think about it.
but it's a good point, and I'll be more worried of your bull passing. Like there's going to be at a 7.5 to 8. I think your 6.5 bull is going to be fine. 6.5 bull is going to be fine. I think he will be your most fertile bull. Most fertile. Well, no, let me rephrase that. From a condition standpoint, he will be ideal at a 6. Between a 7.5 to a 4, I lean towards the 4.5, 5. Um, but yeah,
It's
yeah,
hard to say it though, isn't it?
it is. And again, that's
Yeah.
not the only driver, right? Of
Yeah, exactly. The one thing that fascinates me about your work, and one of the reasons I wanted to have you on, is we always blame the cow, right?
say their
We
rate.
always blame the cow because all
Oh
the
yeah,
sperm's got to do is fertilize the egg, and the bull's responsibility is over.
yeah.
Not the case. I just,
No, no.
I, I find that totally fascinating.
Yeah, and folks in the AI industry, they're noticing that for a few years now. Um, it seems like there's some pregnancy loss driven by bulls as well. There's some bulls that cause more pregnancy loss, and we, as an industry right now, we have no idea why. And by pregnancy loss, I'm talking about cows that we did a day 30 preg check and they're pregnant, and then we run them through at the end of the breeding season, and we run them through at the end of the breeding season, and they lose that pregnancy that made it to 30 days, and they lose differently depending on who they're sire from. So who they're sire by. So it seems like some bulls induce more pregnancy loss as well, which is yeah, it's kind of hard to understand, but it's out there. That variation is out there.
Any more questions from the crew? I'll turn my chat back on. Kevin Laurent, Kevin, I saw you on there. Did you have any comments? Kevin Pedro is one of our beef extension specialists down in Princeton.
No, it's very interesting. I think we all talk about body condition scoring on cows, and again, the bull sometimes is the afterthought. And that this is sounds like a body condition score 5, 5.5 less. Pedro. That might be the sweet spot if we can have an ideal, you know,
I preach
or
nothing over.
maybe a six that you know he's got a little room to lose a little bit of weight while he's getting the job done. I don't know.
Yeah.
I've preached nothing more than a six for quite a while.
Right.
I'm thrilled to see Dr. Fontez's data that, of course, all that was back from Coulter and those guys from Canada for Pete Chenoweth. I know you're familiar with all Pete's work too.
So 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 Les, I guess the question does come to mind. You know, we're in bull sale season. You know,
Exactly.
fat sells. I mean, it
Always.
you know it always does. Whether it's selling bred heifers or bulls. So what about the producer, Pedro, that comes, you know, they buy that bull from that sale right about now. They're going to turn out, say, the 1st of May in our part of the world. Can you give us just a little easy kind of, I know it's kind of a more difficult question as far as depends on the age and whatnot, but give us sort of a warm up period, say for the next 2 months, 6 weeks, 2 months before we turn out on that typical, maybe a little bit at 6.5, 7 bull coming out of a sale. Does that make sense?
Yes, sir, it does. That's a great point. And it really depends on the sale as well. So it's kind of one of the things I recommend is to visit with your seed stock producer about it, about the way those bulls were developed. And and by no means I'm saying that it's wrong to push bulls hard, you know, a little bit during development. That's okay. You know, we want to see the bulls that have genetic merit for post-weaning growth, right? We're interested on that, and that's okay. We start seeing issues when those bulls get extremely overconditioned, like I said. But as you think about transitioning those bulls, very often, and depending on the seed stock producer you're working with, they already started a process of slowing down the amount of concentrate in the diet. They might do maybe some folks we see here, they push them until they have to collect their yearling weight to report to the breed association, and they have the performance data available. Between then and the time they market the bulls, they already start slowing them down, getting the rumen used to a more forage-based diet. So sometimes your seed stock producer might be already doing some of that transition for you. But what I tell folks is, you know, once you bring those bulls home, if possible, try to bring them, you know, more than a month before the beginning of the breeding season. I know that's not always an option, but if you can, that would be good. And then again, those bulls are going to be in a forage-based diet once we turn them out. So if we can transition them to a forage-based diet, predominantly forage-based diet, maybe with a little bit of supplement to keep them gaining. And again, they're depending on the age, they're still growing, right? Most bulls that we're buying or nowadays are, you know, a lot of us are going to the 14, 15-month-old bull type of system or bull sale. We do have some folks selling 2-year-olds as well, you know, but those young bulls are still growing. So we don't want to completely halter their growth once we get home, as we get those bulls home as well. And on that, the other thing is mainly if you're going to turn that bull out in a multi-sire pasture, to commingle that bull with the sires that they're going to be with before turnout. Let them go out there, establish their hierarchy. We know it kind of it's always tricky, right? The day you turn bulls out with the rest of the bulls because you know they're going to fight, and you're just praying that nobody's going to break a leg. But it's important that they establish their hierarchy before you turn them out with the cows, because if you do that, whenever you're turning out with the cows, they're going to fight even more. And when you do that, one recommendations we have is make sure you have enough space so they don't push each other against, you know, a panel or something like. The more space they have to fight and figure themselves out, and before turnout, the lesser the likelihood of them getting injured as well. So those are the two things that we usually tell folks to make that transition a little bit smoother.
Fantastic. Just for those in the audience, remember this is Cape qualified. The Cape code is listed down there, UK Bull. So instead of having your signature from the presenter like normal, just fill that UK Bull Cape code in, and you're willing to get your cost share education from this. And Do you notice that picture there? That picture is where Dr. Bullock is currently. He is at the North Australian Pastoral Company in near Mora, I believe, Australia, where they have tens of thousands of cattle, and he's involved in a 6 week project there, looking at some reproductive heritability traits and reproductive. And it, I told him, he should have brought his repro guy with him, and because Pedro the first day he was there, they had him pulling cedars. I'm like, well, that's not a geneticist's job. I mean, come on, man. But anyhow, Dr. Bullock will be back, and on March 12th, he'll give us a fantastic presentation on bull selection practices. And I know he's going to watch Dr. Fontes's presentation. Again, we really appreciate you coming. A phenomenal job, and data that I know people haven't seen, and so that's what we try to do with this seminars. We try to blow people's hair back every now and then, and you certainly did a good job with that tonight. Appreciate it. And unless there's any other quick questions, we'll see everybody on March the 12th.
Thanks for having me, Dr. Anderson. Appreciate it.